Hey, what's up guys, it's Adam the Science Tutor, and today we're going to be talking about PCR and gel electrophoresis. So, PCR is a polymerase chain reaction, so we're going to need some polymerases, which is in the name. So we're going to use either the TAC, which is TAQ, or PFU. I'm going to be talking about the TAC polymerase more so, but these are going to be heat activated. So think of this polymerase chain reaction as a changing of temperature. That's how we're going to get our DNA to anneal and denature. So the ingredients we need for the PCR reaction is a template DNA. This is our DNA of interest. We're gonna need primers. So these are kind of like our starters. This is what's gonna start our multiplying of our DNA. We're gonna need a nucleotide mix, which is not a big deal. We just need the NTPs, it's usually bought, and a buffer to keep our pH regulated in our reaction, so we have the reaction that we want. So what's the point of PCR? What we're doing, we're trying to multiply the DNA of interest. So in, let's say forensics, like we need to figure out or find see if a DNA matches from a crime scene. We can multiply someone's DNA, specific DNA, and see if it matches. We could do this quickly and cost efficiently with this PCR reaction. So initially we're going to put the ingredients, all our ingredients, into a disc, and we're going to now heat it up. So DNA, as you guys know, is dinucleotide, so they're going to be together. And so how are we going to get this 1 or 3 I think I put in there? How are we going to get these to split? We're going to heat up all of our solution, and it's going to denature these DNA molecules. So we're going to apply heat at about 93 degrees Celsius, and these DNA are going to split. They're going to denature. So it's similar to a protein, similar to different things. If you apply a certain amount of heat, it's going to split these bonds. The van der Waals bonds are going to split. So the polymerase also gets activated, and the primers will attach once these DNA molecules split. If the DNA has GC-rich areas, GC takes a little bit more heat to break than AT-rich areas. So keep that in mind as well. So annealing. So after we split our DNA, we are going to now put it in cold, or we're going to decrease the temperature to about 54 degrees Celsius, and they're going to now anneal and come back together. So, and they're not going to really come back together, but the polymerase, its primary job is going to be to elongate and to build onto these new DNA molecules. But it's all from the same DNA strand, so we're repeating the DNA of interest. So our DNAs will form, and yeah, that's how we're going to get more DNA and the more DNA of our interest. And we're going to repeat the re-annealing, or denaturing and annealing steps multiple times until we have enough DNA that we find sufficient. So this could be repeated about 30 times. And think of our graph. So if we're going to get more and more, it's exponentially multiplies. So as we get more, it's going to exponentially increase in like a graph. It doesn't go 1 by 1, which makes sense because they're each splitting. So this is now the elongation stage. Elongation stage is just where our DNAs are forming, and we're just splitting more and more if we keep doing our denature and annealing stage. And we repeat to have as many DNA molecules of interest that we find sufficient. So we have a lot of our DNAs of interest now, which is good for what we would think we would need. But how do we organize this, or how do we find or see this, and how do we understand what we even have? Well, we're going to have to use gel electrophoresis, and that's going to be what we're going to use to see our DNA and see exactly what it looks like. So once we stop the heat and the cold, or the changing of the temperatures, denaturing and annealing will stop, so we'll stop making more DNA. And we only need to do this about 30 times. We could do this really quick in about a day. We can finish or have like 1,000, I think 1,000,000 copies of coming from 3 DNAs. So you can get a lot of DNA really, really quickly. So like I said, we're going to use gel electrophoresis to see our product and see it and compare it as well. So gel electrophoresis. And what is this? This is going to be a gel, and it's going to be electrons are going to be in this gel, and there's going to be a positive side and a negative side. So keep in mind, DNA has a phosphate attached as a phosphate backbone, so it's going to go opposites attract. It's going to go to the positive side. So we're going to line the markers up at the top, and we're going to fill them with our DNA that we want to be tested, and they will travel down to the positive side because they're attracted. So check out this little diagram thing I drew. So this is like the gel-like substance. So think of it's like threads or like a spider web. So, or cheese threads, I think I put. And so you'll see this, the yellow DNA is very big. So do you think it would go through all that? It would be very difficult because it's so big. It's gonna have a hard time threading through this gel. And then we have our gene of interest. That's gonna have a little bit easier of a time because it's not as big. And then that small pink one is going to be probably the most sufficient at traveling further down our gel electrophoresis. So depending on how you look at this, either last place is the one at the very top, or first place is the one at the very top. It depends on how you look at it. But we'll look at third place. So because it didn't come down further, that means it was very big. So if it on the gel electrophoresis graph, which we'll see right now, if it barely goes down, that means it's a bigger DNA molecule than one that goes all the way down like that pink, because it cannot travel through that silk or that gel. So this is something that you'd probably see, and you're gonna have a key. So our key is to the right, and that gives us about the kilo base pairs. So it's just and there's gonna be reference points to allow us to see where our DNA should be around and where it should be around for different mutations as well. So our DNA of interest, let's say this is that blue one, well, it's going to be, let's say, about 300 kilo base pairs. Then let's say that the red one is a similar strand or the same strand from another individual, or maybe it's on the markers and that's where it should be, and that's about 150 kilo base pairs. So was that a deletion or addition? Okay, that means we added more base pairs. So it was an addition. So maybe we have a mutation that added more base pairs onto that DNA structure, giving us maybe a mutation, maybe it didn't give us a mutation. We'd have to understand. Or maybe it's just a difference in DNA structure from one individual to the next. The green one, as you can see, it's broken up, so that just means it has different points where it was broken. So that would be seen as well, and that's just 3 different areas where you have restrictive enzymes that broke it up in different areas. And then you have the pink, which is obviously going to be the smallest because it's only 10 kilobase pairs, and then the yellow, which is about 900 kilobase pairs. So once again, so for popular or important DNA structures, we'll have keys that will give you a range that your DNA should fall under for a particular gene or particular DNA strand. And if it's off, you can kind of tell if there's a mutation. You could tell with hereditary, or if your family members will have similar sizes in the DNA strand as well. You could pick up on different areas or different markers that show possible disease traits like diabetes or like cystic fibrosis, different ones like that. So you could compare the different DNA sizes, and you could compare if there's an addition or deletion by compared to these markers or the key. Well, anyways, guys, thank you guys for watching and tuning in. If you guys have any questions, leave any comment below. I think I'm just going to go over a little bit more, but if that blue marker, if our strand was longer, we would have a deletion. So cool. Thanks for watching.